going to begin talking about osmosis. But before we do, I want to make sure you're in the right place in your notes. We are actually on page 5 on your notes, and it begins with terms to review. So the two terms that we really need to understand as we move forward talking about osmosis is solvent and solute. Now, a solvent is the dissolving agent, and this is usually water. It can also be lipids as well. We've been discussing how some things are fat soluble versus water soluble. So if the dissolving agent happens to be lipids, then it would be a fat, but in most cases it is water. The other term is a solute. Now, a solute is the substance that is being dissolved. So if we take a look at these two pictures that I have for you, over on the left, oops, sorry about that. Over on the left, we have solutes. These are the things that usually get dissolved in something. So if we want to make hot chocolate, let's say, we're going to take a solvent of water, we're going to add some hot chocolate to it, and then we get a solution of yummy hot chocolate and water. Milk also works too in this particular case. Milk chocolate. The other example over on the right is sort of like salt water. So our solvent would be the water here, and we have the solute as being these little things of salt. So as we move forward, make sure that you understand the difference between solvent and a solute. So we're beginning osmosis, and we've been talking about diffusion, how diffusion works moving substances from a high concentration to a low concentration. Well, osmosis is very specific for water only. So osmosis is the diffusion of water molecules from a high concentration to low concentration. Now, since water molecules are very small, a selectively permeable membrane will allow these molecules through, but it's not going to allow larger ones through. A lot of times it depends on those transport proteins. We do need something like that because it's a polar substance. Now, where there's a lot of water, so we're in a dilute solution, there's a high water potential. Where there is less water, known as a concentrated solution, there's a low water potential. This creates a water potential gradient. So if you imagine what we were talking about for diffusion, how things travel down a concentration gradient, from high concentration to low concentration. You have to think of it in terms of water this time, because water is what is moved in osmosis. So a high concentration of water would have a high water potential, whereas not a lot of water would have a low water potential. So things want to move across water potentials. So when we're talking specifically about osmosis in cells, we have 3 different solutions that we can have. And you're going to see this picture, you're going to see this picture a lot. And just so we're in the right place, check page 6 in your notes, because we've skipped over a question set, and we are now on page 6, osmosis in cells. So the 3 solutions when we're dealing with osmosis, we have a hypotonic solution, isotonic, and hypertonic. They sound the same, but they're going to be different. Hypo, iso, and hyper. So hypotonic solutions have solute concentrations lower than the cell. This causes water to move inside of the cell, as you see in this little picture here. So water is moving inside the cell because there needs to be, basically inside has a low potential, a low water potential. So it needs to move from a high concentration outside to inside. That's where we're moving. Isotonic solutions have solute concentrations equal to the cell. So we have water moving in and out of the cell. This is normal. This is what we really want. We also see hypertonic solutions. Now this means that the solute concentration is higher than the cell. So in this case, water is moving outside of the cell, and we see a lot of shriveled little cells going on. So there's a higher concentration of water inside the cell. It's trying to move out, and unfortunately, the cell is at risk. So in hypotonic, hypertonic solutions, the cell is usually at risk in those particular cases. So, specifically, a hypertonic solution. Water moves out of a cell because the solution outside has fewer H2O molecules. So remember, it moves from a higher concentration to low concentration of water. So this causes the cell to shrink. The cell is going to shrink, it's going to shrivel, and unfortunately, it might die because it's not doing its job. In an isotonic solution, the concentration of water molecules in and out of the cell is equal. So basically, there's no net water movement, meaning that if 2 water molecules move in, then 2 are going to come out. It's equal. This is normal. We really like to be in isotonic solutions. This is where your body works best, in an isotonic solution. Now, if we talk about a hypotonic solution, this is where water moves into a cell because the solution outside has a higher concentration of water molecules. So in this case, we have a higher concentration of water outside the cell. It's moving into the cell, and this causes the cell to enlarge. Now, if it gets too big, then it could actually burst. In the case of plant cells, this is actually good, because this is actually what keeps the plants standing upright in certain cases, like with little flowers and things like that. So if we just do one last review with these, you saw these pictures before. In a hypotonic solution, remember, water moves into the cell, and you're going to see a couple terms. If water moves into the cell might burst. It's called lysing. Or in an animal cell, it's called lysing. Or it might burst, or it's just called lysed. In a plant cell, and this is normal for a plant cell because it creates a turgid environment. This is where you're going to get like a flower stem, or a leaf to actually stand straight. In an isotonic solution, it's normal for an animal cell to be here. Looks normal, acts normal. For a plant cell, it still is okay. However, it might be something called flaccid. It might not stand as straight as it needs to be. It might be kind of a little flappy. And in a hypertonic solution, water moves out of the cell, so it's moving from high inside to low outside. And in an animal cell, it causes a shriveled cell. In a plant cell, the worst case scenario, it could become plasmalized. And this is actually where the plasma membrane is ripped apart from the cell wall, and the cell at this point is pretty much dead. There's no way to fix a cell like this. So if you see a flower or a plant that is dead, basically, it's plasmalized. Chances are the water has rushed out of the cell and it cannot be repaired.